So good afternoon, Ma'am Cindy. So for our reporting, we will be talking about skeletal muscle. We are the group 1, by the way, ma'am. So we will be talking about skeletal muscle, specifically the structure of the skeletal muscle. So for our points for discussion, our objectives, we will be talking about the structure, the fibers and filaments, the sarcomeres, the sarcoplasmic reticulum, A and I bonds, connective tissue covering, transverse tubules, tropomyosin, and troponin. So good afternoon, Ma'am Cindy. So for our skeletal muscle. So skeletal muscle is an organ made up of skeletal muscle fibers, connective tissue coverings, blood vessels, and nerve fibers. So structure of a skeletal muscle. So structure of a skeletal muscle, we have the connective tissue coverings. Each muscle fiber of the skeletal muscle is wrapped in a thin, delicate layer of connective tissue called endomysium. So many muscle fibers are bundled together into groups called fascicles. As you can see in our presentations, you can see there the endomysium and the fascicles. So each fascicle is wrapped in a second layer of connective tissue made of collagen called peromycium. There, the peromycium. Many fascicles are bundled together to form a skeletal muscle. Each skeletal muscle is covered by a third layer of dense fibrous connective tissue called epimysium. So each skeletal muscle is then covered by a fourth tough fibrous layer of connective tissue called deep fascia. So the deep fascia may extend past the length of the muscle and attach that muscle to a bone, cartilage, or muscle. So structure of a skeletal muscle, we have the connective tissue coverings, skeletal muscle fibers. Muscle fibers is a long, thin cell. Each muscle fiber is composed of myofibrils. So each myofibril is composed of 2 protein filaments. So we have the thick filaments, little muscle fibers. Muscle fibers is a thick filaments. The thick filaments are primarily composed of the protein myosin. They have a rod-like tail that terminates in 2 heads, cross bridges. So you can see there in our presentation. And these cross bridges interact with active sites on thin filaments. Thick filaments. So thin filaments, when we say thin filaments, which are primarily composed of the protein actin, they are coiled follicle structures that resemble twisted strands of pearls. So tropomyosin is the rod-shaped protein. You see there the actin. So we have the spiraling around actin to stabilize it. And troponin is a complex of polypeptides. One binds to actin, one that binds to tropomyosin, and one that binds to calcium ions. So both tropomyosin and troponin help control actin's interaction with myosin during contraction. So thin filaments, when we say thin filaments, which are
Striations are caused by the arrangement of thick and thin filaments within the myofibrils. A-band, also known as the dark area. It is an overlapping of thick and thin filaments. I-band, also known as the light area. It is a thin filaments alone. Sarcomeres is the length of each myofibril that is divided into sarcomeres. Sarcomeres meet one another at an area called Z-line or Z-disc. Within the sarcoplasm of a muscle fiber, there are two specialized membranous organelles.
One of the specialized membranous organelles is sarcoplasmic reticulum. It is the network of membranous channel that surrounds each myofibril and runs parallel to it. It is the same with endoplasmic reticulum in other cells. Sarcoplasmic reticulum has a high concentration of a calcium ion compared to sarcoplasm, which is maintained by active transport. Calcium pump, when stimulated by muscles impulses membranes, become more permeable to calcium ions, and calcium diffuses out of the sarcoplasmic reticulum into sarcoplasm.
Transverse tubules. They are a set of membranous channels that extend into the sarcoplasm as invaginations, continuous in muscle cell membrane, the sarcolemma. Transverse tubules are filled with extracellular fluid and extend deep into the cell. Each transverse tubules run between two enlarged portions of sarcoplasmic reticulum, called cisternae. These structures are formed or triad near the region where actin and myosin overlap. Sarcoplasmic reticulum and transverse tubules are involved in activating the muscle tubule. Associated with two sarcoplasmic reticulum, they are termed as triad.